Hi everyone, my name is Eva and my partner is Sia, and today we'll be presenting DNA Microarray assay. Mutations in the DNA of a certain gene may lead to a certain illness or disease. It can be very difficult to detect the mutations since large genomes can have multiple regions where mutations can occur. One method to detect these mutations is using the DNA microarray assay. This is a large-scale genome sequencing technique used to assess whether a mutation in genes is found in the DNA of an organism. It immobilizes thousands of DNA fragments onto a chip. The chip consists of a thin plastic-encased glass disc. On the surface of each chip, there are thousands of small synthetic single-stranded DNA sequences, which together add up to the usual gene in question and to the variants or mutations of that gene present in the human population. This technique is used to measure the expression levels of large numbers of genes simultaneously or to genotype multiple regions of a genome. So how does this technique work? So first, the sample's DNA denatures. This is a mechanism that splits the two complementary DNA strands into a single-stranded molecule. The next step is to break the long DNA strands into a small, more manageable fragments and then apply a fluorescent dye to mark each fragment. Both sets of labeled DNA are then used to insert it into the chip and then allowed to hybridize or bind to the synthetic DNA on the chip. The slide is dried and then it will be scanned to determine how many label cDNA or probe is bound to each target spot. The DNA of the person is labeled with a green dye and then a control DNA will be labeled with a red dye. For example, if a spot turns yellow, then that means that the gene was neither strongly expressed nor strongly repressed in the tumor cells. A black spot will indicate that none of the patient's cDNA has bounded to the DNA in the gene located in that spot. This would also indicate that the gene is inactive. If the individual does not have a gene mutation, all the red and green samples will bind to the chip sequences representing the sequence without the mutation, otherwise known as a normal sequence. However, if the individual has a mutation, the DNA of the individual will not bind appropriately to the DNA sequence onto the chip representing the normal sequence, but it will bind to the sequence representing the mutated DNA on the chip instead.
For the expression profiling, it is useful in compare the expression level among conditions and between health versus disease genes. It will follow the following process. First, it will extract mRNA from cells. The mRNA is extracted from both tissues, the disease and the healthy one. And then they will be reversed, transcribed, and labeled with the dye, usually with the fluorescein as mentioned before. Then they will be hybridized on the array as shown in the figure. Hybridization and washes will be performed under high strict conditions to minimize cross-hybridization between similar genes. The next step is to generate an image using laser-inducted fluorescein imaging as shown on the right image. The principle behind this is that the amount of fluorescein measured in sequence in each specific location is directly proportional to the amount of mRNA with complementary sequence present in the sample analyzed. And the red spot on the array means gene is expressed in tissue 1. The green spot on the array means gene is expressed in tissue 2, and yellow spot means both cDNAs bind and gene is expressed in both tissues. The gray and black one means genes were not expressed in either tissue and they didn't bind either cDNAs. This is an example of the experiment designed to compare the mRNA expressing profile of placentas from women with normal pregnancy and from patients with preeclampsia disease. And the image shows they follow the process of extract, label, hybridize, wash, and scan. So first, they will extract mRNA from the placenta.
It's 10 o'clock.
In panel A, the normal and disease mRNA are labeled with two different dyes mixed and then hybridized on the same array. After washing, the array is scanned at two different wavelengths to yield two images, one for the placenta of a normal patient and one for the placenta of a patient with preeclampsia. In panel B, the single panel, each sample is labeled with the same fluorescein dye, but independently hybridized in different arrays. This is just one example of how the DNA microarray is used, but this technique is immensely useful in a variety of ways. Thank you.
Thank you.